This work was presented at the annual meeting of the American Physical Society, March 4–8, 2019, Boston. Self-organizing structure of protein, CorA, is examined as a function of protein concentration and temperature. CorA protein consists of 351 residues. Residues 1 to 290 constitutes the outer segment and remaining residues 291 to 351 form the inner segment of the protein. Pentamer of CorA proteins embedded in membrane is believed to provide a pathway channel for selective transport of magnesium across the membrane. Crowded membrane is complex to model. Therefore, it would be interesting to investigate self-organizing structures of interacting proteins to assess the effect of protein-protein interaction before attempting to incorporate other constituents. In our coarse-grained model, an amino acid is represented by a solid object such as sphere or cube. The structural details of residue is thus ignored. However, its specificity is captured via unique knowledge-based residue-residue interactions. A number, say, 100 of protein chains are placed randomly in a simulation box. Each residue performs its stochastic movements with the Metropolis algorithm. Simulations are performed for sufficiently long time steps with ample number of independent samples. A number of local and global physical quantities are analyzed. The inner and outer segments of protein chain, for only one protein in simulation box, are found to exhibit different structural response to temperature. Let us look at some characteristics of only one protein chain in simulation box. The snapshots of the inner segment of the protein show conformational consolidation on raising the temperature in native phase. The variations in size of the outer segment does not show a clear trend. The inner segment is found to expand on heating in denatured phase while it contracts on heating in native phase in contrast. The outer segment is less sensitive to thermal response. Analysis of the structure factor provides structural details, that is, the distribution of residues at a range of length scales. The inner segment remained globular while the outer segment exhibits characteristics of a random coil. Let us examine the multi-scale structures of many protein chains in the simulation box. Self-organized structure of interacting proteins show phase-separated globular morphology. Radius of gyration of inner segment increases with the protein concentration at low temperatures, in native phase. Network of entangled proteins seems to appear at high temperature. Variation in pattern of the contact profiles with the temperature reveals how the protein segments organize. Radius of gyration of inner segment increases with the protein concentration at low temperatures in native phase. The radius of gyration of the protein increases at higher temperatures at high protein concentration, possibly due to entanglement. Effective dimension, d, of the collective morphology is estimated from scaling of the structure factor with the wave vector. Effective dimension of the protein, CorA, and its organized assembly is found to depend on the length scale. On smaller length scale comparable to radius of gyration of the protein, the effective dimension is higher than that on large length scale, where the effective dimension depends on the protein concentration. The inner segment of the protein exhibit relatively more variability in estimate of d, which depends on length scale and temperature. Conclusion: stronger protein-protein interactions among the inner segments, CorA, of the protein and its higher structural response show that inner segments play a critical role in a stable self-assembly.